Go, Johnny.
Hello, I'm Johnny. I'm a theoretical physicist by training. I've been a postdoc in the group for the last year-ish. Go Jaguars. Anyway, drug resistance is bad for humans. Could be good depending on what organism's perspective you're taking. But on the flip side, the evolutionary aspects of diseases suggest possibly the ability to control them, given the right tools, steer the evolution to our own ends, to the benefit of humans, to mitigate that resistance, possibly making use of these collateral drug responses that a number of people had mentioned. Kyle had introduced at the top of our group slide deck. But if we really want to make use of the evolution, harness the evolution of disease to our own ends, it takes a lot of information and a lot of resources. And so I broadly want to take my expertise in statistical physics and try to do a lot of very abstract mathematical models that hopefully one day, 100 years from now, will eventually connect to the real world and help people and help study how do we make decisions about how to try to optimally treat evolving disease when you have limited information and limited resources, especially in light of the fact that evolution is very stochastic. We'll talk about the collateral responses, but that's usually there's a whole cohort of possible collateral responses with different possibilities, different probabilities. And so in this big panel on the right, I had studied a really so simplified mathematical model of you have a disease that has two possible genotypes and you're treating it with two possible drugs you can switch back and forth between, and you're trying to optimize to have the lowest possible fitness for the disease, so the best possible treatment success for humans, but using information theory, trying to quantify when you have limited information about what the genotype of the disease is. If you always knew what the genotype of the disease was, you could always give the best possible drug at that moment. But in real life, that would be like biopsying perpetually and then sequencing and getting those results back with perfect accuracy perpetually.
Instantly.
And also having all sorts of data, possibly from in vitro work of how each of those genotypes responds to each drug and all these sorts of things, and putting that all together. And so this is just sort of work that I'm trying to build on now. Again, sort of using statistical physics to say, how can we do our best when we don't know that much? And the plot on the right, the vertical axis is the expected value of fitness in the long term of the disease, so sort of quantifying the success of treatment, and the horizontal axis is the information in the probabilistic entropic information sense, and the bottom line is the literal bottom line that thicker line is saying, as we go along to the right, we can do better, we can minimize our fitness the more information we have, but contrastingly, as we move along to the left and have less information at our disposal, we do worse and can only minimize the fitness of the disease so much. And because this model is so oversimplified, I can actually solve it analytically, which is my favorite thing to do as a theoretical physicist. And so then I can solve for exactly what the equation of that line should be. And what I really love to do is try to bring as much possible analytical solvability as I build up this model into more complex and hopefully more realistic kind of modeling framework. But who knows? It'll probably be computational. But if I can bring any sort of analytical solvability with me, not only will I delight in that, but that will also be useful to streamline computational work as well.
And understand.